Welcome to the NCRI Virtual Showcase. This session's topic is Immunology and Immunotherapy. We are very grateful to Dr. Anna Olsen-Brown of the Clatterbridge Cancer Centre, who has kindly agreed to chair this session and will introduce you to today's speakers. Dr. Olsen-Brown, over to you.
Many thanks and welcome to this session, Immunology and Immunotherapy Ones to Watch. I am a medical oncologist up in Liverpool working at the Clatterbridge Cancer Centre and have a specialist interest in immune checkpoint inhibitors and particularly the toxicities they cause. Just a little bit of housekeeping before we begin. Please do send your questions in for each of the talks. We may take a couple of questions at the end of each talk, but then we'll have a larger Q&A session at the end of the session. There's a Q&A button at the bottom of your screen, and please do post questions as we go along. Please, can I ask you not to take any pictures of any slides which state do not share on them, or which the speaker states not to be shared during their talk? And can I remind you of the Twitter hashtag #NCRIVirtual, and please feel free to use this throughout the talks. So it's now my great pleasure to introduce Maria Semyanikova, who is a PhD student at the ICR, and she's going to be giving her talk on CD4 and CD8 T cells contribute to the efficacy of T cell redirecting antibody CEA-TCB, and TGF-beta dampens its activity in vitro colorectal cancer models. Thank you very much, Maria.
Hi, I hope everyone can hear me. So thank you for the introduction, and I will just jump right in. For some reason, I can't change my slides. Okay, here are my disclosures. So to start off with, I wanted to give a bit of introduction to the immunotherapy landscape in microsatellite stable colorectal cancer, which comprises more than 95% of metastatic colorectal cancer cases. And from now on, I'll refer to it as MSS-CRC or simply CRC for the sake of time. So MSS-CRC have shown poor response to immunotherapies such as immune checkpoint blockade, and that has been attributed to three main factors. The first one being the modest mutation loads. Having these lower mutation loads means that fewer neoantigens are being generated. And actually, our lab has shown that from about 200 predicted neoantigens, only 3 were actually detected by mass spec. And ultimately, that means that there's a lack of tumor-specific T cells. The second factor is the immunosuppressive tumor microenvironment, which can be further subdivided into 2 categories: the acellular factors such as hypoxia and immunosuppressive cytokines such as IL-10 and TGF-beta, and immunosuppressive immune and stromal cells such as regulatory T cells, M2 macrophages, and cancer-associated fibroblasts. And finally, poor infiltration of lymphocytes into tumors, which could be due to either poor T cell recruitment or active T cell exclusion. So a new therapeutic approach that can help overcome the slow immunogenicity in MSS-CRC is the CEA-TCB bispecific antibody, which simultaneously binds CEA overexpressed on the cell surface of many CRC cells, and CD3 on T cells, which triggers T cell activation, proliferation, and secretion of effector molecules. And the advantage of this therapy is that 1, it redirects the T cells directly to the tumor cells, physically bringing them together, and 2, it does not require MHC-presented antigens. However, recent clinical trial data showed that not all patients with CA-positive CRCs respond to this treatment, which means that there might be resistance. So the aim of my project has been to dissect these resistance mechanisms using preclinical immunogenic organoid models, particularly focusing on the role of the immunosuppressive tumor microenvironment. So in order to investigate the contribution of different cytokines and cell populations, I needed a good model. And unfortunately, there are not good mouse models for MSS-CRC because most of them have very high mutation loads and neoantigens. And in addition, they take quite a long time. So as a So we developed an immunocompetent CRC in vitro model, which combines patient-derived organoids, or PDOs, which have been established from multi-drug resistant metastatic CRCs, and T cells, which have been isolated from healthy donor PBMCs. And to make this kind of a simple screening platform as a first step, we seeded the organoids as 2D and added the T cells and the bispecific antibody on top. And the organoids have been GFP labeled, so we can track their growth over time by fluorescent microscopy. And the advantages of this model is that it's fast and it's easily modifiable. So I can add individual factors such as cytokines in different populations and do different combinations specifically in the context of CATCB and see how it affects response. And of course, patient-derived organoids more accurately represent patient tumors than established cell lines. So traditionally, CD8 T cells are considered to be the main cytotoxic effectors, and CD4 T cells are mostly known for their helper role. However, CD4 T cells also express CD3, which means they can be bound and activated by CATCB as well. So I wanted to investigate their contribution to a CATCB-mediated immune response. So in order to investigate that, I used 3 PDO lines that consistently express high CEA and set up a 2D killing assay, which I described on the previous slide. And as we can see here, CD4 T cells can have similar or the same activity as CD8s. So the CD4 curves are here in red. However, not in all PDO lines. So the next question was, what is the main mechanism these CD4 T cells are using for their cytotoxicity? And the main T cell mediated T cell cytotoxicity mechanisms are either the classical perforin granzyme pathway, the interferon gamma pathway, or the Fas death receptor pathway activated by the Fas ligand expressed on T cells. So, sorry, it keeps every time I'm trying to switch slides, it won't let me. So as a first step, I harvested CD8 and CD4 T cells from one of my killing assays and stained them for granzyme. And as we can see here with a control bispecific antibody, which only binds CD3 and not CEA, there is very low granzyme expression levels. However, with the addition of CATCB, we see that there is a massive increase in granzyme B expression in CD8 T cells. However, this increase is very modest in the CD4s. So although this lytic granule mechanism might play a role in their cytotoxicity, it might not be the main one. So next I wanted to look at the role of interferon gamma, and I performed interferon gamma blocking experiments. And although there was 16% lower killing with the interferon gamma blocking antibody in CD8 T cells, there was a much bigger effect at 65% in the CD4 T cells, indicating that interferon gamma might be the main mechanism used by CD4 T cells to control tumor cell growth. And although I'm not showing this data on the slide, the Fas ligand pathway seemed to not play a role in the CATCB response. And from this data, we can conclude that CD4 T cells might be an important biomarker, and that also they might have an advantage over CD8 T cells because they might be able to have a bystander killing effect using interferon gamma, even possibly at low infiltration, even when there's low CD4 infiltration. And additionally, that interferon gamma sensitivity in itself might be a biomarker for CATCB sensitivity. So the next question was, which tumor microenvironment factors cause resistance to CATCB? And the first ones I wanted to look at was hypoxia and IL-10, because in literature they've been described as both immunosuppressive and immunopromoting depending on the context. So it was important to characterize their role in our specific context of CATCB therapy. And neither hypoxia nor IL-10 had any major effect on the CATCB response. So next, I wanted to look at TGF-beta because it has been described to impair T cell activity in various tumors. However, its impact on T-cell redirecting antibodies has not been investigated. And as we can see here, there was a roughly 50% lower percent killing with the addition of TGF-beta, and this effect was even bigger at lower and probably more physiologically relevant E2Ts, with TGF-beta blocking all killing completely at a 1-to-1 E2T or lower in both CD8s and CD4 T cells. And when I was investigating how TGF-beta exerts its immunosuppressive effects, I found that 1, it reduces granzyme B production, and 2, it also reduces proliferation of the T cells that's usually triggered by CATCB. We also have ongoing work with the cellular components of the tumor microenvironment, and this is just some very preliminary data we have from that. So with regulatory T cells, there was only a very small tumor cell growth inhibition effect, although in the future I'll be looking at different CD8 to Treg ratios to see if that has a bigger effect. And paradoxically, the M2 polarized macrophages did not cause resistance and in fact actually promoted killing, but these results still need to be validated in the future. And finally, we also established co-cultures with cancer-associated fibroblasts, seen here in red, and the organoids seen here in green. And just finally, obviously, migration and infiltration was not a component of the first 2D screening system because the PDOs and the T cells are in direct contact with each other. So in order to see how these different tumor microenvironment factors affect migration and infiltration, we increased the complexity of our model and embedded the organoids and the T cells in a 3D matrix. And this is just an example of a time lapse of what that looks over time. So the T cells are here in red and the organoid in green. And here we have a DNA binding death marker, so we can see that with the addition of CATCB, everything, all the cancer cells are killed by T cells over time. And the first question I wanted to ask from this model is whether the size or structure of the organoids affect the ability of T cells to infiltrate. And here are individual slices taken from Z stacks of individual organoids, so these are cutting right through the center of each of these organoids. And as we can see here, the T cells are able to infiltrate into the center of even the really large organoids and also of really densely packed organoids as well. So neither size nor structure seems to prevent the T cells from infiltrating into these organoids. So I hope I've shown you today that both CD8s and CD4s have anti-tumor activity with CATCB treatment. However, the clinical relevance still needs to be determined. I've showed that hypoxia and IL-10 did not impair efficacy in this model, but TGF-beta did, and we're currently investigating combination therapies to counter this immunosuppressive effect. And as I just said, the organoid size and architecture in our 3D model did not impair T cell infiltration. So our hypothesis was that the tumor microenvironment factors are responsible for resistance to CATCB. And we are currently investigating the role of different immune and stromal cells, as well as other acellular factors. Thank you for listening, and here are my acknowledgements.
Thank you very much, Maria. That was really, really interesting. And I just wondered if we could explore a little bit more about that, your 3D model, and whether your next steps are to try and include some of those immunosuppressive elements into the model that you've built so far.
Into the 3D model? Yeah, so that was kind of our workflow where we first wanted to see kind of in the optimal conditions where everything, you know, they're touching, everything is happy to see how that affects. And then we're going to explore their effect in the 3D model as well, because obviously there's the gradient components, the infiltration component, so that's the plan.
Yeah, and I think that's the way, it seems that would be the way to most closely emulate that tumor microenvironment, which has been quite elusive in this field.
Yeah, that's the goal, to make it as kind of closest to mimicking the real tumor, with the ability to, of course, this is still an in vitro model, but the beauty is that we can add individual components and take them away and see exactly what effect that has. So.
Great. Thank you so much. If there are any further questions, we'll come back to them at the end of the session.
Okay, great.
Thank you.
Thank you.
So next I would like to introduce Milana Seraven, and she is a medical oncologist originally based in Spain, but actually working at the ICR currently. And she is going to talk to us about the deconvolution of global gene expression profiles, identifies differently expressed immune-related gene profiles on aromatase inhibitor-resistant estrogen receptor-positive tumours. Thank you very much, Milana.
Hello, can you hear me well?
We can indeed, yes.
Okay. Sorry, I was afraid you wouldn't. Okay, so thank you very much for giving me this opportunity. As Anna has already introduced me, I am a clinical research fellow in Maggie Chang group at the ICR, and I'm going to present some results of our findings regarding the association of some immune-related gene expression profiles with resistance to aromatase inhibitors in estrogen receptor-positive breast cancer. The majority of breast cancers are estrogen receptor-positive, and standard of care includes endocrine therapy, in particular aromatase inhibitors in postmenopausal women. However, 20% of those patients will eventually relapse, and biomarkers to identify those patients at a higher risk are needed. Several mechanisms associated with resistance to aromatase inhibitors include alterations in estrogen receptor pathways, growth factor receptors, and cell cycle, amongst others. Although ER-positive breast cancer is less immunogenic than other subtypes, it is possible that tumor immunity is also involved in resistance to endocrine therapy. Defining mechanisms of resistance to aromatase inhibitors in early breast cancer is difficult because patients are free of disease for long periods of time. Thus, presurgical and neoadjuvant trials facilitate that with the collection of viable paired biopsies from baseline and surgery. I will explain a recent study from Matthew Ellis' group in estrogen receptor-positive, HER2-negative early breast cancer treated with neoadjuvant aromatase inhibitors. In this first graph, they found that some targetable immune checkpoint components such as IDO1, LAG3, interferon gamma, and PD-1 in luminal V tumors are upregulated in endocrine therapy-resistant tumors compared to sensitive tumors measured by higher K67 levels at surgery. They also found a biological correlation of high expression of some of those immune tolerance-related genes with luminal V tumors and poorer prognosis in the METABRIC dataset in ER-positive HER2-negative patients treated with endocrine therapy and/or chemotherapy. In our study, we hypothesized that mechanisms of resistance to aromatase inhibitors in early breast cancer are driven by baseline molecular alterations and changes in response to therapy. We aim to characterize resistant mechanisms to aromatase inhibitors and determine if they are driven by baseline gene expression profiles that lead to response to therapy measured by changes in K67 from baseline to surgery. And in particular, we wanted to evaluate the role of signatures involved in tumor immunity and response to immunotherapy in aromatase inhibitor resistance. For that, we used data from a subset of patients from POETIC, a phase 3 trial that randomized patients 2-to-1 to receive 2 weeks of aromatase inhibitors versus no treatment, and to assess whether perioperative aromatase inhibitors improved outcome compared to standard adjuvant therapy alone. Per biopsies at diagnosis and at surgery were collected. This trial provided validation of changes on expression of K67 after 2 weeks of aromatase inhibitors as indicators of long-term benefit. This Kaplan-Meier is taken from the main report from POETIC published 2 days ago in Lancet Oncology, and Here you can appreciate that patients treated with 2 weeks of aromatase inhibitors, whose Ki67 at baseline is high and remains high, at surgery have a substantially poorer prognosis than those, here you have the red line, with a high baseline Ki67, which is markedly reduced to low. Here there is the overview of our study. We analyzed data from tissue pre and post 2 weeks of perioperative aromatase inhibitors from 118 treated ER-positive, HER2-negative breast cancer patients in which global gene expression profiles were obtained. Part of this data had been reported in breast cancer research. In this sub-study that we are reporting today, we evaluated more specific hypotheses around changes on gene expression patterns and molecular intrinsic subtypes between pre and post-treatment. In this presentation, I will focus on the analysis of 649 modular scores covering different cancer pathways and immune-related genes that we have calculated for each sample. I'll also show the results of differential modular scores expression according to changes in Ki67 between baseline and a 2-week time point. Proliferation rates were estimated as percentage of cancer cells staining for Ki67 and categorized into high, equal or higher than 10%, and low, less than 10% at baseline and at 2 weeks time point. Based on the recent validation of Ki67 a surrogate marker of survival benefit to aromatase inhibitors that I've already explained, we classify tumors into 3 different categories according to changes of Ki67 from baseline to surgery: high, high-low, and low-low. We perform significant analysis of microarray, which is a statistical technique designed to find significant genes or signatures with differential expression values against these 3 categories of Ki67 changes between pre and post-aromatase inhibitor treatment. We categorize tumors into those 3 categories. 28 were high-high, 77 were high-low, and 13 were low-low. Out of these 649 module scores, we found 107 modules differentially expressed across those 3 Ki67 response categories. We performed an unsupervised hierarchical clustering of significant module scores based on their expression at baseline and by Ki67 response categories, and also annotated by intrinsic subtypes at baseline. Our results demonstrated that 29 immune-related modules covering immune tolerance, interferon gamma biology, and response to checkpoint inhibitors were significantly upregulated in high tumors. As expected, we also observed the typical proliferative related features correlated with high samples. And most high tumors were luminal V and included most non-luminal tumors. When focused on the immune-related modules only, it is worth noting that those immune modules track immune checkpoint components and stroma and have previously been captured by TCGA. Those modules include some genes such as IDO1, LAG3, CTL4, STAT1, and interferon gamma. A higher expression of these genes indicates a higher degree of tumor immune tolerance. Interestingly, most of those genes are targetable and have been previously associated with resistance to aromatase inhibitors. I will now highlight one particular module that was differentially upregulated in high tumors and that has been previously associated with response to anti-PD-L1 treatment. Durvalumab is an anti-PD-L1 monoclonal antibody that unlocks the inhibition of the immune response against the tumor. This signature was created to assess response to the anti-PD-L1 checkpoint inhibitor durvalumab in non-small cell lung cancer and urothelial cancer. A high expression of the durvalumab predictive signature was associated with better outcome under anti-PD-L1 treatment in that setting when compared with signature low patients, regardless of their immunohistochemical PD-L1 status. Therefore, an upregulation of this signature in our high patients could indicate a potential benefit from immunotherapy in those early ER-positive, HER2-negative breast cancer patients showing resistance to aromatase inhibitors. Finally, when comparing the changes on gene expression between baseline and after 2 weeks of treatment, we observed similar expression values at both time points in high tumors. Patients were clustered at the same order between these two figures. However, there was a slight downregulation of proliferative related modules in high tumors following 2 weeks of aromatase inhibitors. Based on those findings, it would seem that 2 weeks of aromatase inhibitors do not have a strong impact in those resistant tumors other than proliferation. In closing, we demonstrate that immune-related gene modules are upregulated in breast cancer tumors with early resistance to aromatase inhibitors. These results suggest that high tumor immune tolerance could be associated to early resistance to aromatase inhibitors. And based on these findings, there could be a potential benefit of immune checkpoint component inhibition in this setting that warrants further investigation and that we are already validating in other cohorts in the neoadjuvant setting. I would finally like to thank all POETIC trial participants and funders, the ICR Clinical Trial and Statistics Unit, Dr. Maggie Chang's team, in which I work with, Professor Mitch Dowsett's team, Dr. Saban Dam, and my funders in Spain to support my scholarship. So thank you very much.
Thank you very much, Milana. I was just wondering, in terms of how you view this sort of as a translational process, what would the next steps be beyond validating what you found already?
Yeah, one thing is this validation in other cohorts, but we think that this study is very helpful in terms of maybe establishing a protocol to select the best candidates or target population for clinical trials with immunotherapy in this understudied setting, which is luminal breast cancer, early breast cancer. So this would be, I think, the following steps after the validation. Yeah.
Great, thank you very much. Okay, and then we're going to move on to our third presenter for this session. So I'd like to introduce Hannah Gardner. She is a postdoc at the Netherlands Cancer Institute, and she's going to talk to us about eosinophils are critical for response to immune checkpoint blockade in breast cancer and other cancer types. Thank you very much, Hannah.
Okay, thank you for the nice introduction, and thank you to the organisers for giving me the opportunity to share our work today. As you can see from this slide, a great many people have contributed to this story, so I would like to point out from the beginning that this work is really the result of some great teamwork and a very nice collaboration between the lab of Karen de Visser and the lab of Marleen Kok at the Netherlands Cancer Institute. Oh, now my slides won't move. There we go. Our disclosures. So I think most people appreciate now that immune checkpoint blockade has really revolutionized the treatment landscape of cancer. But for patients with metastatic breast cancer, only around 5 to 10% of them will respond to immune checkpoint blockade. This means that 90 to 95% of patients do not benefit from immune checkpoint blockade, although this can be boosted slightly if ICB is combined with chemotherapy, or if you pre-select for PD-L1 positive tumours, that still only reaches an overall response rate of around 20%, meaning that the vast majority of patients do not benefit from ICB. And I think this really highlights the urgent clinical need that there is to improve our understanding of the mechanisms that underlie response to immune checkpoint blockade, so we can manipulate those mechanisms and make immune checkpoint blockade available to a wider cohort of patients. And with that in mind, we aim to study the systemic immune parameters that are associated with response to immune checkpoint blockade. To do this, we received fresh peripheral blood from patients with metastatic triple-negative breast cancer who were treated with nivolumab. These patients were enrolled at the TONIC trial at the Netherlands Cancer Institute, and we received blood samples at 3 time points: at baseline, after a 2-week induction period where patients received a low-dose chemotherapy induction, and after 3 cycles of nivolumab. And at each of these time points, patients also underwent biopsies. In the interest of time today, I'm only going to show you the comparison of baseline compared to on-nivo, but we have also done the comparison pre-nivo to on-nivo, and actually our results don't really change. So we were really able to exclude any effect of the induction treatment on our outcomes. And with this blood, we performed high-dimensional flow cytometry, and critical to us, we set up a pipeline to analyze the data within 24 hours of blood draw, because we wanted to look at cell types that are often lost through storage, such as neutrophils and eosinophils. So diving straight into the data, here on the left you can see a heat map which is really depicting the dynamics of immune cells after 3 cycles of immune checkpoint blockade compared to baseline. But we wanted to visualize this data in another way to really identify which cells were the most differentially regulated. So we take the same data and visualized it in this volcano plot. So on the x-axis you can see the difference in fold change between responders and non-responders, and on the y-axis you can see the adjusted p-value. And 3 populations emerge as the most differentially regulated immune cells. We have regulatory T cells, CD1c positive dendritic cells, and eosinophils. Now, eosinophils really caught our attention because of recent reports from melanoma studies, and what's been found is that patients treated with immune checkpoint blockade with metastatic melanoma, there is also a correlation to increased eosinophils in the peripheral blood. And this is also, just a bit more about eosinophils, what they are. They're granulocytes, part of the innate immune system. They're packed full of these granules, which contain effector molecules. And in the context of cancer, they're relatively understudied, and despite the fact that they could have potentially a direct cytotoxic effect on tumor cells. Now, they have been generating a bit more interest recently because of what I just told you about results from metastatic melanoma, and it has been suggested that eosinophils could potentially be an on-treatment biomarker. What we see in our data is that eosinophils are significantly upregulated systemically, and here by flow cytometry I show you the fold change in eosinophil counts in responders is higher, and in non-responders it remains the same. And in a pairwise analysis, we also see an increase in eosinophils in responders, not in non-responders. And we validated this with an independent analytical method. So on the left, I show you the flow cytometry data, and here is the hemocytometer counts. And we also made use of RNA sequencing that was carried out on the biopsies taken at the same time points as the blood. And from this RNA-seq data, we looked at eosinophil gene signature, and we saw that the eosinophil gene signature score specifically increased in responding patients and not in non-responding patients. So this really suggests that eosinophils are actually able to infiltrate the metastatic lesions. So next we wanted to investigate whether eosinophils are functionally involved in the response to immune checkpoint blockade, or whether this increase is just a bystander effect. So we made use of a transgenic mouse model. These mice develop spontaneous tumours, mammary tumours, at around 6 to 8 months of age. And what we do is we transplant fragments of these tumours into the mammary glands of wild-type mice, and after which we're really able to mimic the clinical process of metastatic disease. So we perform mastectomy, and after mastectomy the mice go on to develop metastatic disease, and we start treatment with cisplatin and immune checkpoint blockade when mice have established metastatic disease. Again, that mimics the clinic. And what we saw in these mice is that when they were treated with immune checkpoint blockade or immune checkpoint blockade plus cisplatin, eosinophils increased systemically, and we also found an increase of eosinophils in metastatic lesions of mice treated with cisplatin and immune checkpoint blockade. These mice are actually normally completely refractory to immune checkpoint blockade, as you can see from the green line, which almost completely overlaps the control antibody line. So they don't normally respond to immune checkpoint blockade at all. They have a slight survival benefit when we treat them with cisplatin, but interestingly, the combination of cisplatin and immune checkpoint blockade gives them a significant survival benefit. So to investigate whether eosinophils are involved in this response, we depleted eosinophils with an antibody that specifically targets eosinophils, known as anti-Siglec-F. And here you can see the orange line. The therapeutic benefit of immune checkpoint blockade is completely abrogated when we deplete eosinophils. And we know this is specific to the immune checkpoint blockade because we don't, the red line of cisplatin and anti-Siglec-F overlaps the orange line. And finally, we wanted to investigate whether the phenomenon of increased eosinophil counts in peripheral blood is also observed in other cohorts of patients treated with immune checkpoint blockade. We saw the same thing in a cohort of patients with metastatic non-small cell lung carcinoma, where we saw higher fold change of eosinophils in responders and not non-responders. These patients were treated with pembrolizumab. And we also found in a cohort of patients with early stage colon cancer an increase in eosinophils, specifically in responders and not non-responders. But this cohort was interesting because it also contained not only patients with mismatch repair proficient tumors, but mismatch repair deficient tumors as well. And actually the patients with mismatch repair deficient tumors all responded, but we didn't necessarily see an increased fold change in eosinophils in the blood. So we looked in another cohort of patients with mismatch repair deficient tumors, and again we didn't see an increased fold change in eosinophils systemically. So this suggests that eosinophils increase in the blood of patients with mismatch repair proficient tumors, but maybe the increase is not happening in patients with mismatch repair deficient tumors. But we would like to investigate that further. And finally, we also made use of RNA sequencing analysis that was done on this early stage mismatch repair proficient colon cancer tumors. And again, we saw a significant increase in the eosinophil gene signature score in responders, not in non-responders, after immune checkpoint blockade, again suggesting that eosinophils are infiltrating the tumors of these patients. So I hope that I have showed you today that eosinophils are causally involved in the anti-tumor response that is enhanced by immune checkpoint blockade. And I think these data really set the stage for designing novel therapeutic strategies that engage eosinophils to enhance immune checkpoint efficacy in multiple tumor types. And with that, I would like to acknowledge a lot of people. My boss, Karen de Visser, my co-authors on this paper, Olga, Lorenzo and Leonie, our collaborator Marlene Koch, and the rest of our collaborators and all the patients who participate in clinical trials. Thank you, and I'm happy to take any questions.
Thank you, Hannah. That was really interesting. I just wondered, did you have a look at the patient's baseline eosinophil levels, and did you notice any difference, or was it only after a period of time?
Yeah, so there's no difference between responders and non-responders. Their eosinophil counts at baseline is the same, comparable, and it's really specifically to the immune checkpoint blockade that we see this increase.
Okay, and in terms of the postulated mechanism, obviously you inferred that eosinophils could potentially have lots of mechanistic rationale for being effective in this setting. Do you have a particular feeling, or have you looked into what may be the main mechanistic process?
Yeah, that we're really trying to investigate this at the moment. We don't know yet. We're using the mouse model to really look mechanistically what is happening. It's known that eosinophils could be directly involved in anti-tumor killing. They express NK cell associated activation receptors such as NKG2D, so they could potentially directly bind tumor cells, but they could also be involved in an indirect mechanism where they are helping Th2 cells, for example, because they're known to be involved in Th2 responses. So yeah, we're working on that.
The next step. And I just had one other question. Obviously, you've seen a differential effect across the different tumour types. Do you think there's a tumour-specific reason for that?
That's a good question. I don't know. I think what we would like to do is also look in other cohorts to see, is it a universal phenomenon or not? It certainly doesn't seem to be that eosinophils increase in mismatch repair deficient tumours, but that doesn't mean that eosinophils aren't involved in the response, of course. So we would like to look at more cohorts of patients for that.
Okay, great. Thank you very much. So thank you everybody for 3 really fantastic and thought-provoking talks. I think they were all, showed a real breadth of where we next in terms of understanding more about how different immunotherapies work and the potential, particularly into tumour types that we haven't yet got enough or as much as impact as we'd like. There doesn't appear to be any further questions for the 3 of you today, so I'm going to draw this session to a close. If I could just remind everybody that's been listening in to rate the session on the NCRI app using the star rating system. And then obviously there's plenty of other things available to listen to and participate in throughout the next couple of days. So please do go back to the agenda page via the NCRI website for further information regarding the rest of the programme. And it just leaves me to say thank you very much for joining us, and thank you very much for our 3 presenters for really thought-provoking talks.